The overall goal of this experiment is to study the immune responses in the lung during bacterial infection.
This method can help in answering some key questions in the field of lung inflammation, such as what are the key regulatory mechanisms and genes that inhibit or promote lung inflammation. The major advantage of this technique is that it is very efficient and highly reproducible because of the direct delivery of the bacterial inoculum into the lower respiratory tract.
To begin, plate the NTHI on a chocolate agar plate. Incubate the plate upside down overnight at 37 degrees Celsius with 5% carbon dioxide. The following day, culture the bacteria in a brain heart infusion broth. Then, in a microcentrifuge tube, add 1 milliliter of the broth. Spin the mix down at 4,000 Gs for 5 minutes at room temperature. Then, discard the supernatant and resuspend the pellet in 1 milliliter of PBS. Repeat this wash step one more time. Next, dilute 100 microliters of resuspended solution in 900 microliters of PBS and measure the culture's absorbance at 600 nanometers. Use this information to adjust the inoculum to 20 million CFUs per milliliter. Next, determine the viability and CFU